language, it is when you look at the green covering that comes from algae in the water, that means that water is rich in nutrients. And at the end of the day, the oxygen that helps the aquatic environment can also be delivered because of having enriched water bodies. It may not happen in big waters or big surface waters, but if the water is small and the waste is more than even what the water can accommodate, then we end up getting polluted and eventually getting what they call algal blooms and environment where the ecosystem or the aquatic life may be impacted on due to lack of oxygen. But I will not go through all those details. We have got our next part, which is Ms. Aisha Nakanja. And then we know that these webinars happen on the last Friday of every month. So members, if you have something to share with us, to share with the public, to share with the stakeholders, please express your interest through the Ministry of Water and Environment, Water Resources Institute, and email is already seen around, and then we'll be able to schedule to present your findings, a study that informs the sector. Otherwise, I want to welcome Aisha Nakanja to take us through the presentation, and after the presentations, we'll be able to have questions, comments, and suggestions from our audience. Thank you all for attending the 37th webinar of the Water Resources Institute. Thank you. To you, Nakanjo, and members listening. Thank you.
Recording in progress. Good evening. Sorry, good morning, good afternoon, everyone. I'm sorry, I'm having some technical issue with my systems, but I'm trying to resolve it in as much as I can, and I'm coordinating with our host, Mr. Xavier. I'm trying to... Can you hear me?
Yes, we can hear you. However, we are getting some feedback.
I think this is better, and I'll go straight to introduce myself. My name is Aisha. My name is Aisha Nakanja, and I will be presenting today our study that I've shared. The screen is going to be shared here shortly by Mr. Francis. And that said, once again, my name is Aisha. I'm a sociologist and with experience in survey and evaluation research, specializing in water quality monitoring and water resources management. I've worked with the Ministry of Water and Environment and contributed to citizen science initiatives that engage communities in monitoring freshwater ecosystems. I have served as an advisor on tropical freshwater basin catchments through Lake Victoria Basin Advisory Group, and I've peer-reviewed papers for the Citizen Science for Water Conference. I am also currently working with Earthwatch on a project that promotes citizen science for ocean and freshwater literacy among schools. Tonight, I'm going to present to us our study that we carried out in 2023 to 2025. And this was the title of the study. Our presentation today is Citizen Science, Monitoring of Drying Temporary Wetlands, Insights from Freshwater Watch in Uganda and Kenya. We carried out this with our partner organization in Kenya called the Poly Foundation. And basically, this study is going to be looking at the temporary aquatic ecosystems that are prone to climate-driven drying that underpin biodiversity, water security, and livelihoods in East Africa, but remain chronically under-monitored because of their small size, remoteness, and intermittency. Building on the Freshwater Watch citizen science program, this study mobilized over 500 community volunteers in Uganda and Kenya to collect water. Thank you, Mr. Francis. To collect water quality samples from seasonal ponds, streams, and wetlands using simple kits for phosphate, nitrate, and turbidity, along with basic field observations. Members, like I said, between 2023 and 2025, we've worked with volunteers and generated approximately 2,500 measurements across wet and dry phases, substantially increasing substantial and temporal coverage compared with conventional monitoring. Emerging patterns showed that the transitions to low water level and drying are associated with marked nutrient enrichment, visible algal blooms, and loss of perceived water safety for domestic and livestock use, raising clear One Health concerns like linked to contamination in shrinking shared water points. There are also community observations and photographs. These were combined with satellite-derived indicators of surface water persistence to characterize hydroperiod changes and identify hotspots where drying land use pressure and high nutrient loads coincide. So we co-created data dashboards. As a result, we co-created data dashboards and feedback workshops, enabled local stakeholders to interpret results, prioritize repair and restoration, and adjust water use practices using critical low water periods. As a presenter, I'll demonstrate how citizen science can capture the dynamics of temporary waters at scale while embedding local knowledge in management responses. I'll showcase how citizen science offers a practical transferable model for inclusive monitoring and decision-making on drying inland waters in other regions experiencing hydrological change. Members, briefly about the organization Rural Water Initiative for Climate Action. This work was carried out through RICA with support from Earthwatch. And RICA is a legal and RICA in full is Rural Water Initiative for Climate Action, is a legally registered Ugandan non-profit organization governed by a 5-member board of directors and a board of trustees. This structure includes international technical oversight from a One Health advisor to implement system-level solutions for wash, sanitation, and hygiene, and climate resilience through a holistic One Health approach. We leverage academic rigor and practical field experience to build community-scale resilience and ecosystem conservation. Our strategic partnerships are with active technical we are an active technical contributor to the World Water Quality Alliance and the UWASNET, the Uganda Water and Sanitation NGO network, facilitating the bridge between grassroots and data and national policy levels. We use a socio-ecological methodology, utilizing a framework to empower women and marginalized groups as primary decision makers in water stewardship by integrating models, community-led models. We secure the long-term economic and environmental sustainability of rural water systems. And our operational agility, we specialized as a nimble entity. RICA efficiently recollects limited resources to address critical hydrological hotspots where community need and climate vulnerability intersect. As the screen is showing, as I mentioned earlier, I will be presenting in our 37th webinar of the Water Resources Institute. And I'm honored to be here with you this morning to present our findings from our research that was carried out. As you can see on the screen, this is our supporters and partners, World Water Quality Alliance and auspices of the UNEP based in Nairobi, Kenya. And we also work closely with Earthwatch Europe that has supported us with the Freshwater Watch kit that we utilize to disseminate simple, easy to use toolkits that are easy to understand by an everyday person. That we say with citizen scientists. We work with communities, we work with local district natural resource departments, and we disseminate the local the freshwater watch kits with explainable guidelines on how we can use the kits to monitor and look out for parameters in the water samples that we collect. And as you can see in the picture there, also on the map, these are the areas where the Freshwater Watch project has created impact, from Sierra Leone through East Africa to South Africa. The Freshwater Watch and Earthwatch are really doing their best to institutionalize and work to see that citizen science is creating an impact and making a difference in water quality monitoring, because as we all know, water is a very it's a nexus of our environment and a very pertinent resource that we need to conserve and monitor for its health and quality. On the next slide, this is the abstract of our study. It was carried out around Lake Victoria. We looked at temporary drying wetlands around the areas of Lake Victoria and the Mara catchment in Nairobi. And the map above shows the area around the riparian areas around Lake Victoria that we looked at. And again, this study did address the chronic data gap in East Africa's temporary wetlands. We mobilized over 500 volunteers to collect 2,500 data points, capturing hydroperiod dynamics missed by traditional monitoring. Our results reveal a 35% nutrient spike during drying phases, creating a significant One Health risk. By integrating the citizen-led data with satellite indicators, we identified critical hotspots, enabling stakeholders to implement actionable science-based riparian restoration. And if we go on the next slide, we shall briefly, this is the overview of what we are going to be covering: the introduction, the problem, literary review, the theoretical objective, hypothesis, methodology, implementation, results, conclusion, and recommendations. And if we look on the intro, so the temporary wetlands are critical, serving as a primary water source of over 50% of rural livelihoods in East Africa. Our work adopts a One Health approach, addressing the direct intersection of human water security, livestock health, and aquatic biodiversity, of all which are increasingly threatened by climate-driven degradation. If you can move on to the next slide. So with there is the problem is that we looked at temporary wetlands drying, and this is ecological and livelihood significance. Temporary wetlands represent vital hubs for East African biodiversity and local livelihoods. Despite their ecological value, these systems are often neglected in national management and frameworks due to their small size, remote locations, and intermittent nature. And climate-driven degradation, crucially, these wetlands are currently under extreme climate stress as hydroperiods shorten. We observe a concentration effect where evaporation drives hazardous nutrient spikes, primarily nitrate and phosphate exceeding acceptable threshold for domestic and livestock use. The monitoring gap was conventional monitoring programs typically focus on large permanent water bodies, leaving these intermittent systems sometimes under monitored. This creates a data vacuum that masks the degradation occurring in our most vulnerable waters. So we defined the hotspots, and our work specifically focused on key hotspots where these pressures converge: the Lake Victoria Basin, the Nile catchment, and the Mara River Basin. These areas are not just ecological points, they are socio-ecological nodes where changing water quality directly threatens human and animal health. You can move to the next slide. Thank you very much. So we explain here that this analysis synthesizes data from multiple sources, satellite data. We went to areas of Mpigi at Luka landing site, and also we went to Ginger monitoring stations and also the Mara catchment station. And we also had we went to the river gouging stations again in Mara in Nairobi to track the long-term trends of the water levels. And we also looked at data that was a 5-year change period, which refers to the specific intervals used to calculate shifts, allowing us to see how the lakes behaved, how the lake and river behaved in short, manageable timeframes across 5 decades. So we looked at the decadal trends, and we used the meta-analysis results to show how the lake and river levels varied from 1965 to 2010. This was data that we looked at as well. And there was weights of contribution. So there was that different periods had different impacts on the overall model. For example, the period of for example, in the period of 2010 to 2012, this contributed the highest weight at 23.59%, while there was also an earlier trend from 2001 to 2010 period contributed 15.48%. The continuous random effects model, this is our core scientific proof, and state that the model estimate for the lake level was 1134.803 with a highly significant p-value greater than 0.001, indicating that these fluctuations are not random but part of a documented hydrological trend. So we did connect this to temporary wetlands and why it matters. The study explained that these broad fluctuations around Lake Victoria and the Mara River catchment directly impact the invisible temporary wetlands and seasonal ponds we did study. So we also looked at the climate stress when the main lake and river level recedes at an estimated area loss rate of 5.97 kilometers. These smaller fringing wetlands dry up first, leading to hazardous 35% spike in nutrient pollution we identified. Can move on to the next slide. Thank you. When you look at the literary review, we are introducing the theory. We do understand the complex dynamics of drying temporary wetlands. We utilize the socio-ecological systems or the SES framework. This lens is essential because these wetlands are not merely isolated ecological units. They are deeply integrated into the social and economic fabric of the Lake Victoria and Mara River Basin. By applying this framework, we can better analyze how human behaviors such as land use practices and water consumption directly shape environmental outcomes, particularly as systems shift through extreme wet to dry phases. Can move on to the next slide. So here we look at the theoretical model that we used in the study, and we looked at basically the research questions that guided our study. So we had basically three research questions that we looked at, and this did guide our studies towards what we looked at in terms of the research that we set out to study. If we can move to the next slide. And in the objective, we did establish the rationale, which is the degradation of temporary aquatic ecosystems through climate-driven drying anthropogenic inputs presents a growing threat to regional biodiversity and water security. And as well as our study that citing in one of the publications, as Stephen Loisel has extensively argued that traditional monitoring programs often lack the resolution to capture the dynamics of these small intermittent water bodies. Furthermore, citing Mushi's study, recent work on water quality assessment underscores that resolving water use conflicts in regions like the Lake Victoria Basin requires high resolution, community-based monitoring that goes beyond periodic sampling. Our study seeks to address this by moving toward scalable citizen-led frameworks. So defining the objectives, that's how we did define our objective, which was therefore threefold. First, we aim to quantitatively assess how hydro periods, the duration, the water persistence drives nutrient concentrations in temporary systems. Second, we sought to map these dynamics, identifying spatial hotspots where drying intensity and contamination converge. Finally, our core goal was to bridge the gap between data collection and management by empowering local communities to identify high-risk entry points, thereby guiding more targeted evidence-based interventions in riparian restoration and water use adaptation. If maybe we can move to the next slide. And indeed, our hypothesis, as we look at the slide, ladies and gentlemen, our hypothesis was that increased deployment of citizen-led water quality monitoring across climate-sensitive temporary wetlands in Uganda and Kenya will lead to improved One Health outcomes, specifically regarding human water security, livestock health, and aquatic ecosystems and aquatic ecosystem integrity by enabling the precise identification and collaborative management of nutrient and contamination hotspots. As the infographics on the left can clearly demonstrate, this was designed around the Mara River Monitoring Program. And we can move to the next slide. Thank you. So when you look at the methods, the methodology overview and what we used basically, So our analysis of the 2,500 collected samples reveals a strong hydroperiod dependent trend in nutrient concentration. And we did observe a significant 35% increase in both phosphate and nitrate level as the wetland transitioned from wet to dry phases period, providing empirical evidence for the concentration effect in these intermittent systems. And so the data collection sampling that we used, we, like mentioned, focused hydroperiod focused, targeted again sampling of wet to dry phase transitions, spatial density. We looked at sampling scale, hydroperiod focus, spatial density, and data advantage. So with a systematic data collection to quantify the water quality dynamics in these temporary wetlands, we implemented a structured, you can go back to the methods overview. Methods overview. Okay, so to quantify the water quality dynamics in these temporary wetlands, we implemented a structured field sampling program between 2023 and 2025, in contrast to sporadic opportunistic sampling. Our citizen science protocol facilitated over 2,500 individual measurements, systematically captured across distinct wet to dry transitions. Methodological rigor and coverage was that data were collected in a standardized digital format, ensuring high level of data integrity and temporal consistency. This community-led approach provided unprecedented spatial resolution with 10 times the data density achievable through conventional and agency-led monitoring programs. And also the comparative advantage by focusing our sampling efforts specifically on hotspots such as Lake Victoria, the Nile catchment, and the Mara Basin. We successfully captured fine-scale biogeochemical variability that is typically lost in traditional low-frequency monitoring. This dense multi-year data set serves as the foundation for our hotspot analysis and subsequent management modeling. And if we move to the next slide, after the next one. So we looked at the water quality monitoring and citizen science implementation, where we had citizen engagement, national stakeholders at district levels, meeting to identify knowledge gaps, and also communication. We had continuous engagement via online calls and messaging to retain citizen scientists. And this is how basically we maintain communication. Can move to the next slide. Yeah, this was also explained previously on how data was collected, and can move to the next slide. Then when you look at the key results, we see that nutrient dynamics and ecological degradation analysis of the 2,500 collected samples reveal a strong hydroperiod trend in nutrient concentration. And we also looked at ecological responses. This nutrient enrichment is not purely chemical. It manifests ecologically through recurring algal blooms. These were predominantly observed during the dry phases, effectively acting as early warning bioindicators of wetland degradation and impending system collapse. And also the One Health implications, the convergence of these nutrient spikes and bloom conditions creates clear One Health risks, local survey data indicates that during these periods, the perceived and actual water safety for domestic and livestock use is compromised, turning water shared points into potential reservoirs for contamination. And also the synthesis for the finding is that the dense data set derived from our citizen-led monitoring successfully captures these rapid biogeochemical shifts, revealing a scale of degradation that is often invisible to conventional monitoring regimes. And if we move to the next slide, thank you, we see that we are contextualizing the variables that were found. And so beyond regulated outflows and precipitation, we also investigated the role of evaporatization, the combined process of water evaporating from lake surface and river surface and transpiring from surrounding vegetation to see if it significantly drives the fluctuations we observe around Lake Victoria and the Mara catchment levels. The coefficient of our model shows a coefficient of negative 0.017 for evaporatization. This negative value indicates that as evaporatization increases, lake levels tend to decrease, which is what we would physically expect. And the statistical significance, however, when we look at the p-value of 0.251, we find that this effect is not statistically significant at standard confidence levels where p is greater than 0.05. And this suggests that while evaporatization is constant physical factor, it has not been the dominant driver of the major decadal fluctuations in our study period. So as we the key takeaway from this graph and study is that in the broader context of our meta-analysis, this finding is important because it further highlights that human regulated water lake and river outflows, which had a significant p-value of greater than 0.001, have a much more profound statistical impact on lake and river levels than natural climatic loss through evaporatization. And therefore, by showing that variables like river discharge, P equals 0.396 and evaporatization P equals 0.251 are not the primary drivers, we strengthen our argument for why policy focus should remain on outflow management and precipitation monitoring to protect the fringing temporary wetlands. Furthermore, as you look down on the graph below, there is a relationship trend. The bubble plot visualizes the relationship between evaporatization and Lake Victoria and Mara River water levels. The blue regression line shows a slight downward slope, which aligns with a negative coefficient of negative 0.01 we see in our model. Physically, this confirms that as water loss through evaporatization increases, the lake level tends to decrease. So the significance of the bubble size in a meta-analysis bubble plot, this size of each circle represents the weight of that specific data point or study. Larger bubbles indicate data periods that had more influence on our final model results, and the data dispersion of non-significance. While the trend line is downward, you can see significant scatter of the bubbles around the line. This visual dispersion is reflected in our p-value of 0.251. Because the data points do not tightly follow the trend line, we conclude that evaporatization is not a statistically significant primary driver of the decadal fluctuations we are analyzing. Therefore, in synthesis, although evaporatization is a constant natural factor, it is secondary to human regulated factors like river and lake outflows, which showed a much stronger statistical significance of p value smaller than 0.005 in our study. Can move on. to the next slide, please. On the slide of our data curve convergency enhanced by monitoring skills, the methodological innovation was that to move beyond point sampling, we integrated our citizen-led observations with satellite-derived hydroperiod indicators. This allowed us to layer high-resolution field data onto broad-scale spatial trends, and also defining the hotspots. By overlaying satellite data with volunteer records, we identified over 20 critical hotspots. These are the specific locations where extreme drying, nutrient accumulation, and high land use pressure converge. Also, the power of integration. Here, this land use pressure overlay confirms that these hotspots aren't random. They are predictable ecological stress points. This integrates analysis, represents a powerful predictive tool, enabling us to pinpoint exact areas where restoration is most urgently needed. And can move to the next step. So in our outcomes, impact and real-world transferability, we see that we came up with an innovation to co-create dashboards, which were developed to translate the field data into formats that are accessible, interpretable, and directly useful for stakeholders involved in water resource management. By visualizing the spatial distribution of water quality patterns and drying hotspots, the dashboards support evidence-based decision-making and help identify priority areas for intervention. One key implication of the analysis is that riparian restoration should be prioritized in locations where drying intensity, nutrient enrichment, and land use pressure overlap. The project also highlights the importance of locally appropriate adaptations in low water use, particularly used during periods when temporary wetlands are most vulnerable to degradation. And more broadly, this work demonstrates a scalable and transferable model for the monitoring and management of drying inland waters, specifically in regions where conventional monitoring coverage is limited. So in this sense, the approach was not only generate data, but also creating a pathway for operational response, stakeholder engagement, and long-term applicability beyond the current study sites. Can move to the next slide. Can move to the next slide. Thank you so much. Yes, dear members, as we look at our key takeaways and scientific synthesis, is that citizen science captures wetland dynamics. Indeed, our study demonstrated that citizen science is a robust methodological framework for monitoring intermittent aquatic systems. By utilizing freshwater watch kits for grab sampling, we successfully generated high-resolution temporal data, capturing nutrient concentration fluctuations during wet to dry phase transitions that were otherwise invisible to coarser conventional monitoring schemes. This provided a critical evidence base for understanding water quality in remote, small-scale ecosystems. We also looked at actionable hotspots for restoration. The integration of satellite hydroperiod data with citizen-led water quality observations allowed us to pinpoint specific hotspots where land use pressure, nutrient loading, and drying-induced stress converge. While this study did examine these dynamics, we recognize that addressing these pollution pathways requires continuous improvement in the coordination of monitoring. For instance, while nutrient enrichment is a known driver of proliferation in species such as water hyacinths, as seen in sites like Lake Victoria and Mara River catchment sheltered bays, our findings emphasize that pollution dynamics in temporary systems are complex and often site-specific, necessitating targeted evidence-based restoration interventions like riparian buffering. Furthermore, a transferable model worldwide. This can be ultimately, this project establishes a transferable inclusive model for the management of drying inland waters by embedding local knowledge into decision-making through co-created dashboards. we move beyond data collection toward operational management. We recommend that future efforts focus on refining grab sampling protocols to minimize the limitation of discrete sample volumes, ensuring that this scalable framework can be adapted to other climate vulnerable regions experiencing hydrological change. Can we move to the next slide? Dear members, thank you very much for listening. This brings us to the end of our presentation. You've been such a wonderful audience. Thank you very much.
Thank you very much, Aisha, for that presentation and for the work that you did as an institution. We are going to a session of questions and answers. We already have a number of questions in the chat, but as I think we could start with the questions in the chat. I don't know whether you've tried to look at them. Maybe I can read them for you. The first question in the chat, someone would like you to expound on the One Health issue that you mentioned in the presentation, but also on how best they can holistically be addressed.
Thank you, Xavier. On that, that's a good question. So the One Health framework in this research, we looked at the intersection of health. Basically, as we may know, that One Health is basically the interconnection of human health, animal health, and environment health, looking at health as a one health phenomena, and addressing health as a one approach. And so we did explain that our study uses the One Health approach to address the interdependence of human water security, livestock health, and aquatic biodiversity in the Lake Victoria Basin and the Mara catchment. And we did identify shared risks, as mentioned earlier. We found that during the transition to low water levels and drying, there was a 35% spike in nutrient enrichment, phosphates and nitrates, and visceral algal blooms. This creates a shared contamination risk at shared water points used simultaneously for domestic consumption and livestock watering. So with persistent pollutants, our detection of persistent pesticides in some areas, we did look out for these near irrigation schemes, like the Ahero Ahero rice irrigation scheme in Kenya, and also one of our sites in Kalangala highlighted some traces of toxicology risks that travels through the food chain from aquatic life to humans if there is pollution in the water streams, and this is a One Health risk. So the practical application and how it was used, again, using the socio-ecological monitoring by utilizing the SES, socio-ecological systems framework, we analyzed how resource scarcity during drying phases alters human and livestock behavior around wetlands. And also citizen-led hotspot mapping. Citizen science allowed us to map over 20 hotspots where land use intensity, hydrological intermittency, and contamination overlap. This data is more relevant to health outcomes than traditional monitoring because it captures the exact points where people and animals interact with stressed water. And the benefits could be adaptation and mitigation, informed restoration. Going forward, adaptation of this model allows national agencies like the Ministry of Water and Environment to prioritize riparian restoration in specific hotspots that pose the highest One Health risks. And also the preventative management, there can be mitigation approaches, which can now be scientific-based, maybe rather than reactive. For example, the co-created dashboards help local stakeholders adjust water use practices during critical low water periods to avoid pathogen and nutrient spikes. Also, there can be scalability. So this research provides transferable models for inclusive decision-making It demonstrates that empowering communities to monitor their own resources is the most suitable way to manage the health of climate-stressed intermittent freshwater ecosystems globally. So again, that question was very good. And so basically One Health turned our study from a water quality report from community survival strategy by identifying a 35% nutrient spike during drying phases. We did provide the evidence needed to protect both the livestock and livelihoods depend on, and the domestic water security over 50% of the rural population in this region uses.
Yes, thank you.
Wow.
Thank you, Aisha. There was another question. This is from Bridget. Bridget wanted to know what informed and guided the selection of the hotspots that you carried out during the study.
Yeah, thank you, Bridget. That's another great question. If you asked about our data and the method or the motivation for the hotspots, how they were selected, basically the role of citizen science, I can provide a comprehensive approach highlighting three main areas: scale, frequency, and integration. So with data providing, where did the data come from? For example, the primary field data, we mobilized over 500 community volunteers in Uganda and Kenya who collected approximately 2,500 water quality measurements. Also standardized kits, volunteers used professional Freshwater Watch kits to measure specific chemical parameters, including phosphates, nitrates, and turbidity. There was also meta-analysis. We synthesized historical satellite data and local monitoring records from the historical as far as 1965 to 2010, and also coming closer to 2023 to 2025 to establish the long-term decadal fluctuations around Lake Victoria and the Mara catchment. But also citizen science methods helped, and explaining this, we can just look at it as a support tool. It was essential for reasons such as capturing temporal dynamics. Traditional monitoring sometimes looks at bigger water catchments, but small or remote intermittent water, unlike small and remote intermittent waters. So citizen science allowed us to capture high frequency data during critical wet to dry transitions that would otherwise have been invisible. And also this increased spatial scale. By engaging local water users, we achieved 10 times the data coverage compared to traditional monitoring methods. This allowed us to map over 20 specific pollution hotspots where land use pressures and drying overlap. Lastly, coming to that questions, there was embedding the local knowledge. Citizen science allowed us to combine professional chemical testing with community observations and photographs. This embedded indigenous stewardship directly into the management responses. I don't know if I should go further, but maybe I can close that very question with the validation rigor. In case you ask about the quality of the citizen data, we can look into the validation, which is comparison with the professional labs. We cross-checked referenced suspected pollution points by taking triplicate samples and transporting them to the Water Resource Institute laboratory in Entebbe, as some pictures did show us for professional nutrient analysis using standard methods. Also, satellite integration, we integrated volunteer observation with satellite-derived indicators of surface water persistence to ensure the ground level data matched larger hydrological trends. Yeah.
Thank you, Aisha. There is another question from Michael. Michael is saying turbidity is very high according to your analysis. Did you try to find out why, especially during the dry period? Maybe let me go through them. like 5 questions, then you answer at once. Is it okay with you?
Yes.
Yes, I think that will be better. So that was a question from Michael asking about turbidity being high, especially during the dry season. Then there was also another question from Wanja. Wanja was saying, with this information be shared with the community in the study area in a simplified format, something which is easier which is not scientific and technical, just for the layman to understand. What's the plan of sharing the data with the water resources management and also the wetlands management department and any other relevant institution? Then also another question from Abdallah Matovu, do you have any other parameters collected other than the mentioned one, especially the physiochemical parameters? Then we have a question from Ambrose. We need to have this information shared with us. We need this information. Then Annette is saying, is there any publication you can refer the team to? Then there was another question. What are the practical interventions that can be implemented at the district or sub-county level to tackle the changes in small catchments? So basically, those are the questions we have in the chat. You can attempt them as we wait for other questions from members. Members, feel free to raise your hand in case of any clarification or question. Thank you. Back to you, Aisha.
Thank you, Xavier. That was a good question on turbidity being high compared in dry season. So again, because it was a time scale period, it enabled us to look out for these parameters even during dry and wet seasons. The concentration effect, which we looked at the evaporative concentration, while it may seem counterintuitive, since there is no rain to wash the sediment into the water, turbidity spikes during the dry season due to the concentration effect. As temporary wetlands recede and water volume decreases, the ratio of suspended solids solids to water increases significantly. And therefore, our data from over 2,500 measurements show that the transitions to low water levels are associated with marked nutrient enrichment and visible algal blooms, which did contribute to high organic turbidity. But also, there was the anthropogenic and livestock pressure, which is the One Health link. So during the dry seasons, these temporary wetlands become the primary water sources for over 50% of rural livelihoods and their livestock. So the increased physical activity specifically livestock, trampling at shared water points disturbs the bottom sediments, keeping them suspended in the shrinking water columns. So this aligns with the SES, the Socio-Ecologic Systems Framework, which analyzes the human and animal behavior shifts during resource scarcity to shape environmental outcomes. But I may also want to talk about the Mara River Monitoring Program. In our specialized monitoring of the Mara River Basin, we recorded that 78% of samples showed evidence of elevated sediments runoff NTU are greater than 40. We found that turbidity was elevated in nearly all sites, but intensified during low level periods where sediment becomes more visible and concentrated. I may also want to maybe talk about the phytoplankton and the algal growth. So the 35% spike in nutrient enrichment, the phosphate and nitrates, we did identify during drying phases often triggers harmful algal blooms. These cyanobacteria's blossoms increase the biogenetic turbidity of the water, making it appear cloudier and safer for domestic use. So maybe with that, I have answered a bit of that question. And with the question if we have any other parameters collected. Mainly, we did look at our Freshwater Watch kits looks at 3 parameters: the nutrients, phosphate, nitrates, and turbidity. But with more equipments and we can definitely be able to look at more nutrients. But for this one, we specifically looked at only those nitrates, only those parameters. But the study did utilize a specific set of chemical and physical parameters to monitor water quality across the temporary wetlands in Uganda and Kenya. Yeah, so that having said, the chemical parameters again were phosphate, nitrate, also nitrogenous compounds, and pesticide residues. Yeah. So another question was that what are the practical interventions that we can look at the district level in small catchments? So based on our research findings and outcomes from the Freshwater Watch support program, practical interventions at the district level for managing small catchments could include community-led restoration and buffer zone and restoration of buffer zones. So targeted riparian restoration districts should prioritize, for example, the restoration of riparian vegetation specifically in identified hotspots where land use, intensity, and high nutrient loads coincide. We can also look at the protection of buffer zones, which is implementing enforcing buffer zones around small water bodies, which helps filter agricultural runoff containing phosphate, nitrates, and pesticides before they enter the water systems. We can also look at inclusive monitoring and decision making. We can look at One Health mitigation approaches. If maybe time allows, I can briefly talk about this. For example, adoption of citizen science. When you look at inclusive monitoring, districts can formalize citizen science programs to bridge the chronic data gap in under-monitored remote intermittent wetlands. Also, data-driven prioritization. We can use the co-created interactive dashboards and volunteer-generated maps to identify high-risk areas for rational allocation of scarce management resources. Lastly, on the One Health mitigation approaches, we can look at management of shared water points. Because drying wetlands lead to a 35% spike in nutrient enrichment, as we observed, district could manage these sites as shared water points to prevent contamination risks for both human, domestic, and livestock health. I don't know if time can allow, but I would also like to talk maybe briefly about the adaptive water use. Districts can lead feedback workshops to help stakeholders adjust water use practices during critical low water periods when pollutants are most concentrated. Otherwise, if there's another question, yeah, that. I hope I've answered that question.
Okay, thank you, Aisha. Do we still have other questions? Members, if you have any question, raise your hand. Yeah, Aisha, there was a question which I think you must have missed. There was a question concerning the hotspots. What were the determining factors for you to choose? For example, you talked about a landing site in Impigi and also other hotspots. What did you consider when you're choosing these hotspots? Is there anything specific that made them special?
We looked at areas, riparian areas around the Lake Victoria and the Mara catchment, and we looked at the remote areas next to the wetlands that are exposed to drying, and that was one of the criteria for mapping the hotspots.
Okay, thank you. Then there was a question on I think also you must have missed it. Is there any publication that you could share with the team?
Oh, yes.
That
Yes, that's a good question, Annette. And we definitely will share the publications with the Water Resource Institute team and the presentation as well. But yeah, there are publications you can definitely share, and I'm going to definitely share with you, Mr. Xavier. Yeah.
Okay. There's a question, there's a hand, there's a hand from Ambrose. Ambrose, please come in.
Yes, thank you for this presentation. I would like to understand, because we have seen a hotspot around the Lake Victoria, but when we see the hydrology, it starts from the low areas. So which plan do we have to maintain good quality of water in these areas? Because water is flowing from these small systems to big systems.
Yes, Ambrose, that's another good question and observation that we did start from the low catchment to the high catchments, and what measures can we put in place to ensure good water quality in all the from the lower to the from the low catchment to the higher grounds. So to ensure good water quality in small catchments where hydrology begins in low-lying areas. For example, you can have a plan. A plan must integrate upstream landscape management with localized community stewardship. While pollutants, for example, often concentrate in low flow areas or hotspots, the source of these pollutants is frequently linked to broader hydrological and land use patterns across the catchment. So again, that's a very good question. Based on our research, there can be a multi-level plan to achieve good water quality. For example, landscape scale mitigation can be the source to sink plan. Since pollutants usually like persistent, for example, pesticide and fertilizers, or these are carried downstream by rivers into the low-lying lake basins management. And so must start, for example, at the highlands. So if we establish riparian buffer zones, that can be a good plan, or manage high pressure agriculture, that can be another good plan. Also specialized monitoring of temporary waters. Can look at One Health mitigation approaches, like I explained. There can be governance and district level implementation, and so on and so forth. If time allowed, I could maybe break down further, but yeah, I don't know if Francis can guide us on that. Otherwise, yeah, that was a good question.
Aisha, I think we still have some time. You can break it down for the benefit of the participants.
Great. So, Ambrose, when you look at landscape scale mitigation, which can be source to sink plan, when we look at establishing riparian buffer zones, we can target identified entry points of restoration by planting native vegetation along seasonal streams and wetland edges. These buffers act as biological filters for agriculture runoff containing nitrates and phosphates. However, we can also look into managing high pressure agriculture, which is implementing strict oversight near large-scale irrigation schemes such as rice and palm oil plantations, where persistent agrochemicals, for example, insecticides and fungicides, are more frequently detected. When you look into specialized monitoring of temporary waters, temporary water bodies are often invisible, sometimes to larger monitoring or national monitoring programs, but are critical to rural water security. So if we capture wet to dry dynamics, utilize citizen science to provide high frequency data during the transition from wet phases to drying phases. This is crucial because drying leads to 35, like we saw, lead to a 35% spike in nutrient enrichment, turning these areas into toxic hotspots. So we can also look into integrating community knowledge, combining local observations and photographs with satellite-derived indicators of surface water persistent to create real-time understanding of hydroperiod changes. When you look into One Health mitigation approaches, these are plans that I'm breaking down for us. Water quality management must address the intersection of human, animal, and ecosystem health. So managing shared water points during drying phases, shrinking water bodies become shared domestic and livestock points. Plans must include strategies to implement contamination from livestock, waste, and pathogen concentration in these shared spaces. And we can also look into addressing pathogen ammonia spikes. So when we use parameters like ammonia to detect recent pollution and total nitrogen to monitor long-term degradation, that can be a plan. And when you look into a plan of governance and district level implementation, we can, for example, look into data-driven prioritization, using co-created interactive dashboards like earlier presented to help district stakeholders interpret results and prioritize scientific restoration in most critical areas. Secondly, adaptive water use practices, lead feedback workshops to help local communities adjust water use habits during critical low water periods and avoid health risks associated with visible algal blooms and nutrient spikes. I remember when we started, Madam Gwendolyn did mention about the issue of algal blooms and nutrient spikes on water sources. So if we carry out these kind of plans, it can help us assess better and even mitigate better on monitoring our water quality, but also the agile institutional stewardship, leveraging nimble organizations, for example, RICA, Rural Water Initiative for Climate Action, and all other organizations where we are all from to bridge the gap between grassroots data collection and national water quality policy management. So by addressing the socio-ecological feedback loops, where resource scarcity alters human behavior, these plans move beyond reactive monitoring to inclusive, preventive, and water stewardship.
Thank you very much, Aisha. Do we still have any question or clarification that is needed? Feel free to type in the chat or raise your hand. I feel and I think the presentation has been very interactive. I want to thank Aisha for the presentation, for the research that they did. Since we don't have any other question, I want to invite the coordinator, Water Resource Institute, to give us the closing remarks for this webinar. Thank you very much.
Mr. Francis?
Yes, Aisha.
I think I saw a question from Nathan, Mr. Nathan Sechita. I don't know if I answered that. How did you ensure data quality consistency across more than 500 volunteers? Since Mr. Francis had already invited our dear host, Madam Gwendolyn, I can briefly just take you through about this. briefly. Nathan? Mr. Francis, is that okay? Do we have time?
Yes, it's okay. It's okay. Go ahead.
Okay, so Nathan, that was another good question. So we're looking at high quality, which is, by the way, a good concern with citizen science. So to ensure a high data quality and scientific consistency, for example, across the 500 volunteers that we did mobilize with varying literacy levels, we did implement rigorous multi-layered quality assurance frameworks. This approach, of course, moved beyond simple data collection to creating a system of trusted science through methods such as standardized protocols and low literacy tools. For example, the Freshwater Watch app is a very easy to use app. The use of dedicated smartphone applications streamline the process. The app uses, because the Freshwater Watch kit has a Freshwater Watch app, and the app uses icons and guided steps to ensure that users, even with low literacy, can accurately input data. And also visual documentation. Volunteers were required to take smartphone photos of the water bodies and test results. These photographic vouchers do allow our technical team to manually verify the conditions reported on the ground. Also, there is continuous training and engagement. We do capacity building. We did just hand out kits. We did conduct district level stakeholder meetings to identify knowledge gaps and train coordinators. We also have feedback loops, which is continuous communication, which was maintained through online calls, messaging, and emails. This established an environment of trust and allowed for real-time troubleshooting if a volunteer was unsure of a procedure. Also, training stewardships with regular engagement ensured that volunteers remain committed to the protocol. We also had scientific validation and triangulation, integrated analysis, and so with all that, we were able to have quality data from our citizen scientists, and that's what I can say. Nathan, I hope I answered your question.
Okay. Just hello, ladies and gentlemen, and they had called me earlier to thank our presenter and also to thank you participants that have joined us today to listen in to Aisha's research and study that has been carried out between Uganda and Kenya and other countries, and how impactful the research can be to form that decision, to form how we can approach to management of the water and environment, especially where it comes to water resources management. Can you hear me? And want to
Yes, we
thank
can hear you.
Aisha. Want to thank everybody that attended today's webinar. And I want also to call on you to come in also and present your findings, present your study, present your research. It cannot be the research we are looking at does not only be the scientific research. You can have a practical approach, you can have policy research, you can have policy studies, and all these ones are welcome to be part of the Water Resources Institute webinar, which is a monthly webinar series. I remember there's a time when we didn't have monthly, especially the time when we were approaching the Uganda Water and Environment Week. We went through about 5 webinars that were aligned to the Water and Environment Week themes and sub-theme. In case there are more people who want to present, and we find that we can easily make it even more than one webinar in a month, but that one depends on the demand. So when the demand is high, the supply will be able to supply you with the time to share your knowledge, to share your findings with our communities that are mainly underwater and environment sector, and even beyond Uganda, beyond our region. Thank you, Aisha, and want to say thank you to everybody, and we say bye and have a blessed weekend.
Goodbye.
Okay.
Bye.
Bye-bye.